heard there was something twisting the head and moving away from the microphone, but make some kind of note if you don't hear me anymore. So thank you. It's a true pleasure being here in Japan for the first time. So thank you, Sofion, for the very generous invitation. I really appreciate that. So this talk, as we just heard, will be a switch of targets. So I will talk about a potassium channel within the Kv7 family. And we will be looking at the cardiac subtype, Kv7.1, which together with the hERG channel is important for cardiomyocyte repolarization. And Kv7.1 forms a typical tetrameric structure, like other voltage-gated potassium channels. It has a voltage sensing domain that couples to the pore domain and opens the gate upon depolarizing stimuli. And in cardiomyocytes, Kv7.1 is co-expressed with an auxiliary subunit called casein E1, which forms a single transmembrane segment. And this together is the Kv7.1 casein E1 channel, also called the ICS channel. So my comfort zone is usually in lipid modulation of this channel. So we have been working on how different type of lipids interact with the voltage sensing domain to enhance channel function. But now we did a long trip to beautiful Japan. So I thought I would be brave enough to move a few angstroms in the channel. So we will go from the voltage sensing domain and to the pore domain and look at some of the ongoing work we're doing on how steroid hormones like estrogens interact with the channel to inhibit channel function. And we are not the first to be interested in steroid hormone modulation of ion channels. This has been studied for many ion channel targets by many different groups. And this type of modulation is quite complicated because it could involve many different mechanisms of action. So for instance, steroid hormones like estrogens, they could signal indirectly on ion channels through different hormone receptors. There could also be genomic pathways and also direct ion channel effects through direct interaction with the ion channel proteins in the membrane. And this is just one example from the literature, and it's for the BK channel, and just showing the complexity of hormone modulation of ion channels. So we are interested in steroid hormones for two reasons. One is that we have a broad interest in how compounds, endogenous compounds, but also exogenous compounds that have this type of steroid scaffold, how these molecules interact with Kv7 channels, which Kv7 channels are targeted, and what would be the functional effects. And this is kind of a new interest in our group. Another interest, or another reason for studying the steroid hormones is our collaborations with clinicians, in particular cardiologists, because it's known for a long time that women in general have a longer action potential duration and QT interval than men do. And there are many reasons contributing to this, but one that has been highlighted is that steroid hormones have effects on the ion channels of the cardiomyocytes. And for us, it's particularly interesting that the native cardiac current IKS, which is generated by the Kv7.1 casein E1 channel, it's been reported in many studies before to be inhibited by the main female sex hormone estradiol, or E2 for short. And we would like to understand how that inhibition happens. So today I will present some of the work we've done or are doing in an ongoing team effort to understand estrogen hormone interactions with Kv7.1 casein E1. And a lot of the slides will be on preliminary unpublished data, so please tell us what we're doing right and what we're doing wrong so we can understand this better. But we will go, so in this collaboration, we go from synthetic chemistry to molecular dynamics and also manual electrophysiology in my group. We are collaborating with Nina, who is in the audience, for automated patch clamp electrophysiology. And we collaborate with cardiologist Katja Odening at Bern University in Switzerland for the genetics of long QT syndrome or cardiac arrhythmias, and also different rabbit models. And it's particularly fun to present this here at this meeting because it's one of the first projects we get a chance to try the Q-patch. And as a Star Wars fan, I like to think of the Q-patch as Sophion's own R2-D2. So you see, there are similarities between them. Okay, so what are the functional effects of estrogens on the Kv7.1 casein E1 channel? Well, so these are examples of experiments from xenopus oocytes expressing the human Kv7.1 casein E1 channel. And these are what the potassium currents look like under control conditions. They get bigger and bigger in response to stronger depolarizing stimuli, and we can close the channel with a hyperpolarizing pulse. If we acutely apply estradiol, or E2, to these oocytes, you see that the current gets smaller. And this is a general reduction in current amplitude. You see examples of here without changing the voltage dependence of channel activation. So the inhibition, the magnitude of the inhibition looks similar, no matter if we quantify what happens at the maximum conductance or at voltages that are more relevant to the cardiac action potential. So I will use these quantifications a bit interchangeable because they are all the same. It's a general reduction. This is what the concentration response curve looks like. And this is from the xenopus oocytes expressing the human channel. And we see fairly similar inhibitory responses following a concentration-dependent effect, also on mammalian CHO cells expressing the human Kv7.1 casein E1 channel. So estradiol reduces these currents without changing the voltage dependence. And this is very similar to what has been described in the past in different systems by other groups, that estradiol inhibits this channel and the native IKS current. So it's nice that we can recapitulate what's already been described. And we also see similar effects in different models that we've been working with. For instance, in rabbit cardiomyocytes, estradiol is inhibiting the native current IKS, which is generated by the Kv7.1 casein E1 channel. So this inhibition that we see, is it through direct effects on the channel or indirect effects involving any of these signaling pathways that hormones can use? And this is a tricky question to answer because there are so many signaling pathways for hormones. But what we see, for instance, is that the inhibition is quite fast. So this is for xenopus oocytes, where it takes about 10 minutes to get saturating effects. And this is faster than what we would expect if genomic pathways were involved. We can also repeat these experiments, but in the presence of inhibitors of the main indirect signaling pathways that are used by estrogens. For instance, we can inhibit the estrogen receptors, but also G-protein coupled receptors that are used by estrogens. And even if we inhibit these signaling pathways, we see the same magnitude of channel inhibitions as we do under control conditions. So although these experiments don't rule out indirect effect, we think that they at least open up for the possibility of direct effect, since we see similar effect even if we inhibit the main indirect signaling pathways that can be used by hormones. So if there are direct effects, from where could they happen? And there are many residues to look for. So to have some guidance, we used molecular dynamic simulations in which we allow estrogen hormone molecules here in green to explore the channel and try to find their kind of preferred sites of interaction. So occasionally, you see that some of these hormone molecules will stay longer at certain positions at the channel to form longer-lived interactions. So this is kind of a guidance to our experiments and where to start looking. And if we quantify where it's most likely to find the estrogen hormone molecules on the channel in these simulations, we find a putative interaction site over here. And this is in between the Kv7.1 subunit, and you see the S6 segment in pink, and the auxiliary subunit casein E1, which is labeled in blue. And if we resolve this putative interaction in more detail, you see that the estrogen hormone seems to be close to serine 338 and phenylalanine 335 on the S6 segment, and serine 64 on the casein E1 subunit. So in experiments, we can follow up on these predictions and mutate these residues to see if this change the estradiol effect, yes or no. So this is a way of telling if we're on the right track or not. And here you see experimental data, and this is for a estradiol concentration that for the wild-type channel normally gives roughly 60% current inhibition. But if we mutate any of these potential interacting residues, you see that the magnitude of the inhibition is much reduced. So mutating predicted interacting residues impairs the effect of estradiol. We can also be a bit more harsh. We can do the same experiments, but we remove the whole casein E1 subunit, which we believe is part of this interaction. And just for guidance, this is casein E1 is predicted to adopt approximately the same position as the casein E3 subunit, here resolved in the structure from Sand and McKinnon. So if we now do the very same experiments, but on Kv7.1 alone, so lacking the casein E1 subunit, there is no effect whatsoever of estradiol. So there is no reduction in the current, no matter how high we go in concentration. Estradiol has no effect on the channel if the auxiliary subunit casein E1 is not there. So we need casein E1 to get this inhibitory effect. We can also look at the hormone molecules. So instead of mutating the channel and the putative interacting residues, we can also mutate chemically the hormone by looking at hormones with different chemical properties. And one thing that was striking in our simulations is that it seems like the two OH groups of the estrogen hormones form hydrogen bonds with these two serines, and potentially aromatic interactions with this phenylalanine. So to focus on these OH groups that we think may be important, we compare the effect of three synthetic estrogens that share some of the kind of core geometry of estradiol, but vary in their OH group. So two of these hormones have OH groups at approximately the same positioning as estradiol has, whereas the third hormone is lacking these OH groups. And what we see in the experiments, both in xenopus oocytes and in the Q-patch assay, is that it's only the two hormones that have the two OH group similar to estradiol. It's only those hormones that also have the inhibitory effect. If we have a hormone that's quite similar in the core but lacking the OH groups, there is no inhibiting effect anymore. So suggesting that these OH group may play a role in the interaction. And we are now going back and revisiting the natural hormones that are related to estradiol, because these are interesting too, because some of them have one OH group, some have some other changes to where the OH groups are positioned, and this is still ongoing work, but what it looks like in preliminary data is that estradiol is by far the best channel inhibitor, and those hormones that lack OH group only have one OH group, either have no inhibitory effect or smaller effects, but we're still following up on this. So as a final note, we find it interesting that the casein E1 subunit, it has many disease-associated mutations that are just near this serine that we predict would be interacting with estrogen hormones. So for instance, we have, starting with position 67, which is just one helical turn down from the serine 64, and then following, all of these are arrhythmia-associated mutations linked to long QT syndrome. So since they are so close to where we think there might be an estradiol binding site, we wonder whether these disease-associated mutations may change the effect of estradiol or not. So to test this, we exposed each of these disease-associated mutations to a low or a higher concentration of estradiol. And we find that some of the mutants, like this one and these two, they respond to estradiol with clear current reduction or channel inhibition, and with a magnitude that matches what we see for the wild-type channel for the low and the high concentration, which is indicated by the dashed line. But this is in contrast to some of the long QT mutants, like these two that show an impaired response, and these two long QT mutants that don't respond at all to estradiol. So just to highlight what it looks like, of the responding long QT or disease-associated mutants, some follow a wild-type-like response, whereas some mutants no longer are sensitive to estradiol. So it seems like just out of these arrhythmia-associated mutants that we have tested, we can kind of divide the mutants into those that are good responders to estradiol, just like the wild-type channel, so those would be inhibited by estrogens, whereas there are kind of bad responders that are no longer sensitive, or at least less sensitive than the wild-type channel. Okay, so to sum up, we found that estradiol inhibits the Kv7.1 casein E1 channel, and that the casein E1 subunit is required for this effect. And the inhibition of this channel, both in heterology systems but also in native systems, is similar to what others have been described. For instance, there is a beautiful paper from University of Tokyo on this in guinea pig cardiomyocytes. So we recapitulate what others have seen, and we see similar effects throughout different experimental models. What we do find is that there are diverse responses of mutants and of disease-associated mutants that people are carrier of, and that there are diverse responses also of related steroid hormones. So our working hypothesis is that there might be a direct effect of these hormones on the Kv7.1 casein E1 channel, and that this may come from a putative binding site just at the interface between the Kv7.1 subunit and the casein E1 subunit, and that the chemistry of the hormone, and especially OH groups able to form hydrogen bonds with specific residues in this pocket would determine the effect. And that's why we think that if you carry long QT mutations in this area, you might deform or change this pocket, and this is why you might see differences in the estrogen effects depending on your genetic background. So, ongoing project, many open questions. For instance, if this is an estradiol binding site, we wonder why estradiol binding there would reduce the current. So this is something we would like to understand. We would also like to be able to rationalize the impact of certain mutations, so why some are abolishing the estradiol effect and why others don't change the estradiol effect. And the same for hormones, the native or natural hormones we are now revisiting. We're curious on if other compounds that have this type of cholesterol scaffold is sharing this pocket, and of course, is there any clinical role of the effects that we see? That's something we're interested in. And this last one it's important to us, but it's tricky to address because xenopus oocytes, CHO cells, or even rabid cardiomyocytes, they are not humans, and hormones have so many signaling pathways and targets in the human heart. So we think we provide insights, but how to link this to the clinical context, it's trickier. A final limitation is that we are kind of struggling with how to interpret the concentrations of hormones that are needed in different experimental systems. So if anyone can advise us on this, we would be super happy. Physiologically, you could have picomolar and up to hundreds of nanomolar of estrogens during certain circumstances, but that's still lower than what we need to get prominent effects in many of our experimental systems. So of course, the concentrations in acute experiments differ from physiological concentrations. And here, it seems to matter a lot to us if we use plastic in our experiments, what solvent we use for estrogens and so on. So any advice on that, we would be happy kind of to learn. With this, I would like to acknowledge the people in my team and previous people who have been involved in starting up this steroid hormone research line that we're also doing, all our collaborators and the funding, and to Sofion for giving us the possibility to be here at this meeting. And if you, in the autumn, get low in your dose of ion channels and you want to travel, you should find Nina and discuss visiting the Ion Channel Conference in Linköping in Sweden. So with this, thank you for your attention.
Thank you, Sara, for super clear-cut presentation. So any questions? Yes.
Thank you, Sara. Great talk. So I'm wondering if the stoichiometry of KCNQ1, KCN1 affects the amount of inhibition. So, or 4 KCNE1 are required to inhibit the channel or not.
Yeah, that's a great question. I'm not sure I put that as a backup slide, so I don't have it here, but the stoichiometry matters. So we have tried linked constructs with different stoichiometry. So if you have 2 casein E1 in the channel, there is less estradiol effect, or it's a shifted dose-response relationship of the estradiol effect compared to if you have 4 casein E1 subunits. So the fewer casein E1 subunits you have, the less effect of estradiol there is. And I think this is interesting from a cardiac perspective, where it's still a bit discussed, the stoichiometry.
Thank you.
So, good question. Thank you.
Thank you.
Yes, please.
Hi. So I have 2 MD questions, one from molecular dynamics, one for medical doctor stuff. So the first one is that you show the binding site for estradiol, and the serine 64 that you show looks quite far from the pocket. Have you tried running it in all atoms and show that there is a hydrogen, actually a hydrogen bond between the OH group and the serine? And the second MD, which is the medical doctorate, is that do you see the long QT syndrome mutation on the E1 associated with the gender? Because estradiol is supposed to be like female sex hormone. Do you see any association with that?
Super good MD questions. For the first one, yes, we have followed up with all atom simulations. And actually what I showed you here was for one of the synthetic hormones, but we've done exactly the same for the natural hormone. And you're right, serine 64 in this specific snapshot is a bit far away, but the hormone is really exploring this interaction. So there are hydrogen bonds forming with both of these serines and also at the same time.
Brilliant.
For the doctor MD, that's one of the things we collaborate with Katja on, to look back at the clinical data to see for which mutants, long QT mutants, there is a clear sex difference in the arrhythmias, and if there are others, there is not a clear link to sex. That would be very interesting to compare for these mutants. Unfortunately, we cannot do it for these mutants because they are very, very rare. So these are long QT5 mutants. There are only a few cases per mutant. So we don't have the kind of statistics and the material for that. But we will go back now and try to pick long QT mutants on the Kv7.1 side, which are more common, to see if we can look at sex differences in those. Because in general, for long QT mutants, there is a sex difference where females have more severe symptoms and are more prone to arrhythmias like this, but we can't tell for each specific mutant what it looks like.
Thank you very much. Really interesting.
Yes. Good questions.
Okay, one last question, Jamie.
Hi, Sarah. Very, very nice talk. I just to follow up that question, there is evidence that women are more susceptible to long QT syndrome. And is there any mutation that are known that are associated, which has more severe symptom in women than in men? Do you know that, or?
Yeah, there are. I mean, in general, women with long QT1 have more severe symptoms and are more prone, but that's kind of collecting all the mutants together. But there are some mutants that are more common. I can't get the number out from my head, but I can check it up. And for those that are more common, and we have enough kind of statistics material, women are kind of have an even longer QT interval than men do. And this is something they have during, after puberty and before menopause. So somehow it's related to the sex hormone, but it could be several mechanisms. So we would like to compare one such mutant with one we predict would not be affected by estradiol to see if there is no sex difference.
Right. So another comment is that so that the binding site where you found that's very critical for the coupling between Wald sensor and the pore opening in channel in general, so that it's not only necessarily just the casein U1 mutations, but some casein Q1 mutations probably will also be affected, or be the effect that could be associated with casein Q1 mutations. That may not be necessarily in that particular spot, but is in general important for the coupling between Wald sensor and the pore.
Yeah. Yeah, that's true. And it might be that estradiol, if estradiol binds there, it's kind of interfering with the coupling, so shutting off kind of the communication pathway, potentially. Yeah. But coupling is difficult, as you know. You're the expert on that.
Okay. Sara, thank you very much.